All right, everyone. Good morning. Um, so, yeah, good morning. I do want to address one thing before we get started. I do apologize for all the craziness with the grades with exam one. So I think we have it all figured out. Unfortunately, I still think I can't make everyone happy with this particular scenario, but I think we've worked it out where this is the best solution. So here's what happened. Again, I tried that Canvas curve function for the very first time. I will never use it again, by the way. Okay, so what I did last night is I reverted everyone's score back to the raw score, all right? But then this morning, I added 6 points across the board to everyone's score. So if you haven't looked at the grade book yet, you will actually see exam one curve. That's what the title is called. It's an assignment out of 0 points, and then everyone across the board got 6 points. All right. So that is the curve. That's the curve for both section 1 and section 2. Every single person got 6 points. Unfortunately, there are some students that were at the lower end of the curve that received more points because what happened with that curve function was it brought people up from the bottom end, but it dropped people from the top end. So basically, if you got a 98, all of a sudden you got an 88. Okay, so I couldn't let that stand. I made sure that no one lost any points, but then I had to go back and just start all over again. All right, so this is how the curve will work from now on. You will get a raw score, and then right next to it will be added points. I like to see the average between 76 and 78. That's where I shoot for every exam. Okay, so if some semesters I have a 72, then everyone will get about 5 points. Okay, any questions? All right, so I guess the group of individuals that really I feel the most upset about, again, this was very stressful for me too, are those that lost points with the second curve. Those were the students that were kind of at the lower end. I will, to really address that, I will be giving another extra credit opportunity the week after we get back from spring break. That will be an additional 5 points just to really help with that issue, okay? So that, just make sure you're here that day because it's kind of another kind of competition that you'll turn in a sheet. It's going to be the Tuesday, March 17th when we get back, all right? Any other questions that we have at this point? All right, so going forward, it should go a lot more smoothly, and again, I won't use that function again. All right, so we are recording at this point. What we're going to be doing today is we're going to finish up with skeletal muscle dynamics. We left off last time talking about smooth muscle cells. Is that correct? Smooth muscle cells. We did get through that particular diagram. So now today I'm going to just do some comparing and contrasting of all types of muscle, whether it's skeletal muscle, cardiac muscle, or smooth muscle. So we're going to do a little bit of exercise. Just so you know, too, Meghna did a really good job of actually grading your assignments here. They are in alphabetical order. You can come up after class and grab them on the way out. And if I forget to say this, have a wonderful spring break when you're leaving. Okay, be safe, and yeah, boy, did it come up fast. Okay, so let's go ahead and get started. We will have a top hat question, but it's going to be later on in the lecture. All right. Also, you have received problem set number 6. So I will be going through some equations today to really help you with that. And I'll give you a little time today to kind of work on it as well. All right. So I'm going to go up to where we left off. Here's where we left off, correct? We actually went through this particular cellular mechanism, the sequence of events that occurs with smooth muscle contraction. So just to review, this actually starts where we get an elevated amount of intracellular calcium. A lot of times this is coming from internal stores, although the internal stores are a lot more minimal within smooth muscle compared to skeletal muscle. So a lot of the calcium comes from the extracellular fluid, or if there is some kind of GQ coupled receptor, it's more of a minimal sarcoplasmic reticulum, SR. Okay, so here's where we start off. You get an increase in cytosolic calcium. This is the same for all three muscle types. Okay, all three muscle types, contraction occurs when intracellular calcium rises. All right, in this case though, calcium activates an enzyme called calmodulin. All right, the calcium calmodulin activated molecule then goes on to activate myosin light chain kinase. Those are proteins that are situated on the myosin neck. If you go back and you take a look at what a myosin molecule looks like, it has a tail region, a head region, and a neck region. Okay? So the myosin light chains are actually situated right on that neck. All right. Calmodulin activates myosin light chain kinase. Kinases phosphorylate proteins. And the activated myosin light chain kinase actually phosphorylates the neck region of the myosin molecule. That causes the myosin head to be more on a 90-degree angle. You can actually see this right now, right here on the right-hand side. Then when that occurs, basically myosin and actin can interact. So smooth muscle doesn't have troponin, doesn't have tropomyosin. All of the regulation is right there in the myosin neck, okay? So once it's phosphorylated and the myosin head can now interact with actin, cross-bridge cycling can occur, just like we've talked about before. Okay, it ratchets, basically those myosin head ratchets the actin together, and the muscle contracts in all directions. Usually it's wrapped around a vessel or a tube like the small intestines. Okay? All right. Once calcium is either resequestered in the internal stores or removed across the sarcolemma, and intracellular calcium wanes, decreases, what happens is the myosin light chain phosphatase takes over, its activity takes over, and it dephosphorylates the myosin light chains. And when that happens, the myosin head actually is held near the thick filament, and now it can't interact, and the smooth muscle relaxes. Okay. All right, so this is an important slide. You need to know those sequence of events. Do know what calmodulin is, myosin light chain kinase, and myosin light chain phosphatase. It's not short answer. Just make sure that you recognize the players with this sequence of events. All right, so let's go ahead and keep going. We're almost done with this particular section. If you want to take a look at this flow chart when you get home, I think this is a great study aid. What are some of, that previous slide was really comparing the differences between smooth muscle and skeletal muscle, okay? Here's some of the inputs that actually influence smooth muscle contractility or contractile activity. Smooth muscle can actually have spontaneous rhythmic contractions. That's what happens in the small intestines, right? When you're talking about motility in the gut and moving a bolus of food down the line. Basically, a lot of times in the small intestines, for instance, there's a type of motility called segmentation, where one part of the small intestines is relaxed, the other one is contracted, and it goes back and forth like this. This is actually called segmentation. It actually helps to mix and move the food down the small intestines. So this rhythmic, spontaneous activity is due to those smooth muscle cells that surround that particular tube. It's interesting with this type of spontaneous, depolarization. It looks something like this. These smooth muscle cells are actually expressing the same channels that pacemaker cells have in the heart. So that rhythmic myogenic type of depolarization occurs because of the expression of those funny channels. And we'll get to that after spring break. But you can see it's not a nice, steady resting membrane potential. Because of the expression of those funny channels, it's an eroding depolarization, a slow eroding depolarization that reaches threshold and then activates a calcium channel. You get these calcium spikes, right? That causes a rhythmic depolarization and contraction with these smooth muscle cells, okay? All right, another input, going back to the previous slide, are neurotransmitters. A good example of this is the sympathetic nervous system. If you get really scared, what happens is smooth muscle cells constrict blood vessels that are going to your extremities. It diverts blood to more vital organs like your brain or your heart if you have to run or think of some kind of escape path, right? So it's diverting blood basically to more vital organs, and these are more prolonged contractions, all right? That's why your face or your hands may turn white when you get scared, right? It's diverting blood to other organs. So neurotransmitters released by the autonomic nervous system also play a role in smooth muscle contraction. Hormones also. We'll be talking about these hormones when we get to the renal section, but there are hormones like vasopressin. Just like the name suggests, it's smooth muscle cells that press around blood vessels, vasopressin, and angiotensin, right? Angiotensin is another hormone that causes vasoconstriction. Okay. Also, locally induced changes are important. These are more intrinsic mechanisms. If you have a local environment that's high in carbon dioxide, obviously this part of the tissue or this part of the body is not getting sufficient oxygen. And that elevated carbon dioxide levels actually tend to dilate, relax smooth muscle cells, okay? So there's local changes within the environment that surround smooth muscle cells that can also cause contraction or relaxation. And then stretch. This is an auto-regulatory mechanism. If you have a surge of high blood pressure, it's going to stretch those vessels and the smooth muscle cells, and then there's going to be a compensatory constriction to keep flow fairly constant. All right? So again, back to homeostasis. That stretch can also cause mechanical channels, mechanical gated channels to open and then cause constriction, compensatory constriction. All right, do you all have any questions about that so far? All right, so these are just, again, some inputs that influence smooth muscle contraction. All right, this slide is giving you basically some information between single unit and multi-unit smooth muscle cells. Smooth muscle cells. So I'm going to blow up the top one right there first. The top one is the single unit, and the bottom one is the multi-unit. And I'll show you what you're looking for, basically. Here's the single unit smooth muscle cells. Just as the name implies, all of these smooth muscle cells actually contract at the exact same time. So again, they contract in unison. And the reason why they can do that is because they actually have gap junctions. If you take a look at each of these cells, do you see where my arrow is? You can see that gap junctions are actually connecting all of the smooth muscle cells, okay? So when that calcium floods into the intracellular space, it actually, that calcium wave goes from cell to cell to cell very quickly so that all of the smooth muscle cells contract in unison with each other, like a single unit, all right? Multi-unit are different because they don't have gap junctions. So they contract independently with each other. Pretty good? So without gap junctions, then it just depends on when that neuron, that motor neuron, is fired, or that autonomic nervous system, all those neurotransmitters are released. And they will contract independently from each other. That's why this is called multi-unit. All right. So let's move on away from smooth muscle cells. Just remember that there's some key differences, but all muscle requires an increase in intracellular calcium for contraction to occur. Yeah?
What would be an example of a place in the body where smooth muscle would need to be coordinated?
Yeah, I would say that a lot of times with blood vessels, that would be the case. You actually want, one smooth muscle fiber around a vessel probably wouldn't give you that restriction and flow that you need. So you probably have a lot of different smooth muscle cells that need to coordinate that contraction. Yeah. And that would be true with the intestines too, actually. Yeah. All right. Let's go ahead and now talk about the differences between skeletal and cardiac before we move into the cardiac section. Okay? All right, so these are two really important terms that help you kind of distinguish between cardiac muscle and skeletal muscle. All right? So the top part of this slide is actually referring to cardiac muscle in particular. The term myogenic applies to cardiac muscle because basically what it's saying is the depolarization that occurs. Remember that electrical current has to sweep through the muscles first and then contraction can occur. That first depolarization that occurs in the pacemaker cells is inherent to the muscle itself. All right, again, as I mentioned before, these pacemaker cells are actually expressing those funny channels. They're called HCN channels, or hyperpolarization cyclic nucleotide gated channels. But I think it's easier for you to remember that these are funny channels, okay? And again, you have this eroding depolarization that causes the cell to just reach threshold all on its own, and then you get further depolarization that basically sweeps through the entire heart, and then contraction can occur to propel blood out, okay? So this genic is referring to the origin of that electrical stimulus, and myo is referring to the muscle. So it originates in the muscle itself. Let me just make sure that everyone is aware of this. You don't need any neurons. You don't need any hormones. These pacemaker cells depolarize all on their own. Okay? That's what allows for your heartbeat to continue on a regular basis. Yeah?
So if that's the initial depolarization is myogenic, can it still be modulated from the top to increase the heart rate?
Yes, that's a really good question. So it is the autonomic nervous system that basically is innervating those pacemaker cells. Neurons are actually physically interacting with the pacemaker cells. So when you get scared, it releases norepinephrine and it increases the cardiac pacing, or the parasympathetic does the opposite. So yes, it can be modulated by the autonomic nervous system. Yeah, great question. And we will be getting into more detail on that. Yep. All right. Now, the only reason why the depolarization occurs in the pacemaker cells first, the only reason is because it depolarizes the fastest. That's the only reason. It depolarizes the fastest, and then that depolarization wave moves through gap junctions from cell to cell to cell. And it's those funny channels that actually give those cells an unstable resting membrane potential. Or some people call it an eroding depolarization. All right, so what happens when something goes wrong with the pacemaker cells? This actually happens to some patients. This is actually known as sick sinus syndrome. Sick sinus syndrome. And you probably, I don't know if you remember Dick Cheney, or anybody know Marshall Stern in our department? Dr. Stern. If something like that happens, then Medtronic is a big company that actually makes these pacemakers, right? What you have to do is they implant a pacemaker into your body, and you don't even have to have the electrode to the pacemaker cells. It can be anywhere in the atrium, but it takes over as the pacemaker, okay? So, interesting. All right, remember skeletal muscle cells are neurogenic. Okay, we've already talked about this pretty extensively. For skeletal muscle to contract, you have to activate a motor neuron, right? That's basically how skeletal muscle contracts. So with that, skeletal muscle is neurogenic because that depolarization wave comes from neurons, motor neurons. So neuro is referring to the neurons. Genic is the origin of the electrical stimulus. So it can have multiple or single twitch innervation sites. Okay? All right. Here's another really important difference between cardiac and skeletal muscle. We've already talked about this before. This is kind of a nice slide to illustrate that, though. Cardiac muscle, this is really the interaction between dihydropyridine receptors and ryanodine receptors. Okay? As we've talked about before, the dihydropyridine receptors are the voltage-gated calcium channels. So they sense those action potentials traveling down the T-tubules. That change in membrane voltage actually does activate these dihydropyridine receptors, which are voltage-gated calcium channels. Calcium floods into the intracellular space, and then it's calcium that activates the ryanodine receptors. Now you can imagine that this is a positive feedback mechanism. The more calcium that floods into the intracellular space, the more ryanodine receptors are activated, the more calcium floods from the sarcoplasmic reticulum. It's just a way to amplify that response. This is known as calcium-induced calcium release. All right, with skeletal muscle, it's a little bit different though. We've already talked about this as well. With skeletal muscle, the dihydropyridine receptors and the ryanodine receptors are actually physically interacting with each other. So when the action potentials propagate along the sarcolemma, the dihydropyridine receptors, they do detect that change in membrane voltage, and there is a conformational change, but it doesn't conduct calcium. So calcium doesn't flow through that voltage-gated calcium channel. It still can detect the change in membrane voltage, but that conformational change actually is translated to a conformational change of the ryanodine receptors because, again, they're physically interacting with each other. And that opens up the ryanodine receptors and calcium floods into the intracellular space. So this is called depolarization-induced calcium release. All right, pretty good? All right, so continuing on, just a little bit more about skeletal muscle. Skeletal muscle is striated, just like, cardiac muscle. This is cardiac muscle. Cardiac muscle is striated, just like skeletal muscle. So it is organized in sarcomeres. It does have troponin and tropomyosin, and they are fused cells. So there's 1 or 2 nuclei within each muscle fiber. But I think the structure that you're really looking for cardiac muscle is the intercalated discs. Intercalated discs. And if you look closely at the bottom here, right? If you look closely at the bottom, There are intercalated discs are made up of desmosomes. I don't know if you remember that from epithelial cell section, but these are those spot welds that help the fibers kind of stick together, stay together. The intercalated discs are where all the gap junctions are as well. And there's a number of ion channels that are expressed within that intercalated disc. So you can see gap junctions right here as well. And that's kind of one of the defining features of cardiac muscle. All right, here's another difference that I think is really fascinating. Take a look at the top panel is actually what the action potential looks like in skeletal muscle. All right, the action potential is in green, and there's hardly an hyperpolarization, as we've talked about before. It's only about 5 milliseconds in duration. And then again, remember, you always have to have the electrical current first and then contraction. So the orange line is basically contraction. This is an increase in force generation, and then relaxation. Now, take a look at cardiac muscle. This is the action potential in cardiac muscle within the ventricles, ventricular myocytes. Okay? You can see the action potential. There's a rapid depolarization and then a very long plateau phase that lasts as long as the contraction itself. And then it repolarizes. The duration, instead of 5 milliseconds, is 250 milliseconds. That's a huge difference. All right, take a look at this. That causes the refractory period to be as long as the contraction itself in the heart. All right, so first, I want you to think about all 4 relationships that we were talking about earlier. Intensity force, frequency force, length tension, or load velocity. Which of these relationships is going to be affected by this prolonged refractory period? You can talk to your neighbors. Let me know what you think. First, you have to remember what refractory period is. And then you have to think about which relationship is going to actually be affected. Whenever you feel like you have the answer, just go ahead and raise your hand. Don't be shy. Yes. What do you think?
We thought it was frequency force.
That's right. Okay. It is the frequency force relationship. Okay, so do you want to venture as to why you think it's frequency force?
Maybe to keep keep a certain time between each contraction.
That's right. That's really the essence of it, right? Okay, so the refractory period, let's just review what that definition is. The refractory period is, especially absolute refractory period, means that no matter how strong a second stimulus is, you cannot fire a second action potential. So it prevents the firing of a second action potential. So just by definition, you can't increase the frequency, right? Until basically the cell repolarizes. So this prevents a second action potential in this 250 millisecond period of time. All right? Why would that be important? What does it prevent ultimately? Think about that frequency-force relationship. What is it preventing? Yes. Yes, exactly. So think about that. It is preventing summation and tetanus. The last thing you want your heart to do is cramp up, right? It's preventing summation and tetanus. So that's a really important feature of cardiac muscle. These are actually calcium channels that open up and stay open for a while, keeping the cell depolarized for 250 milliseconds. That's called the plateau phase. All right, very important in the heart. All right, so if you need some text associated with what we've been talking about, this is actually a really good summary. And this one you can use when you get home. This is actually a table that is comparing and contrasting skeletal muscle, cardiac muscle on the very right-hand side, and smooth muscle, single unit and multi-unit. Okay, so it's really a nice compilation of everything that we've been discussing. Okay? All right, so I will let you take a look at that at home. What I want to do now is, if you wouldn't mind getting your top hat devices out, let's go ahead and start with a top hat question with some of the material that we just covered. I got to switch over to section 2. All right, so let's see here. Okay. All right, so we just have one for now. Oops. And then I'll have one after I lecture on it's going to be more of an intro to circulatory system. So we actually did talk about this. What proteins are expressed in the intercalated discs of cardiac muscle? A, desmosomes, B, ion channels, C, gap junctions, or D, all of the above. This should be a pretty easy one, unless you fell asleep for those 5 minutes. We talked about that. Intercalated discs. All right. Got about 45 students that have answered already. Let's go ahead and give you 10 more seconds. Anybody need a little more time in this room? All right. So I'm going to go ahead and close this, and then, of course, the correct answer is D. All right. Yes, if you did answer A, B, and C, that is correct, right? All three of these are expressed in the intercalated disc, but the best answer is D, all of the above. All right? Okay, so I'll go ahead and close this one up. We may have one more depending on how far we get. I will continue to put the top hat questions. Megna has been great about getting all of your homework assignments in, and I think our exam is now kind of completed. So everybody should have most of their grades entered. I think the only thing left is the length constant extra credit. So I will be continuing to put those in as well. All right. So what I'd like to do now, and unfortunately this is tough for people that are watching the video at home, right? I'm going to be drawing on the board right now. What I'd like you to do is get out your problem set number 6. And let's start with some of the essential cardiovascular calculations, okay? All right, so I'm going to, it's more of a little chalk talk. We're going to take a break for a minute. And we're just going to talk a little bit about some terms that are going to be important. I think for some students that may be working in the clinics, maybe either a vet office or medical scribe or whatever, you probably heard a lot of these terms, and sometimes it's really fun to put these things together. But I still can't assume that everybody knows these terms. So I'm going to go through it fairly slowly. And then I'm going to return and talk about Ohm's law again, okay? So let's start by taking a look at this. This is your essential cardiovascular calculation sheet. And so there's a number of different equations. Some of them are going to be given to you, and I'll tell you which ones. In fact, that equation number 2 already on your website is really what's going to be stapled to the back of your paper copy, or it's going to be provided for you for the online copy. All right. So let's start off with what is systolic pressure and what is diastolic pressure. Okay? I think that's one of the first things that you need to understand. All right. First of all, systole. I'm actually writing that on the board now for those that are at home. Systole is a term that we use to describe the contraction of the ventricles. Systole is basically when the ventricles are completely contracted. All right? So we're talking specifically about ventricles. Contracted or in the process of contracting. It's called systole. Diastole is when the ventricles are relaxed. So again, it's specifically referring to the ventricles. Systole is basically when it is contracted, diastole when it's relaxed. All right, so we'll learn about this a little bit later as well. Think about your blood pressure. Usually when you go to the doctor, they actually put on a blood pressure cuff called a sphygmomanometer. We'll learn more about that later. But basically it is measuring the pressure in your brachial artery. All right? So when your ventricles are completely contracted, it propels your blood into that brachial artery, and the pressure there is going to be the highest. It's called the systolic pressure. When your ventricles are relaxed, that's when the pressure in the brachial artery is going to be the lowest, your diastolic pressure. All right, so you probably have heard this before. 120 over 80. That's your blood pressure. You go to the doctor, they say, oh, it's good, 120 over 80. So now I'm going to give you a little bit more information. The units for this is millimeters of mercury. That's a unit of pressure. Millimeters of mercury. And the top number is the systolic pressure. This is what happens when your ventricles contract and propel blood into that brachial artery. That's when the pressure is going to be the highest. The bottom number is the diastolic pressure. That's when the ventricles are relaxed and the pressure in the brachial artery is going to be the lowest. Now on your sheet here, it's actually looking at the systolic and diastolic pressure of the aorta. All right, so it's just another way of measuring, again, when the different areas, when the ventricles are completely contracted or relaxed. Okay? All right, so now I want to kind of give you really the essence of this particular cardiovascular calculation worksheet. It all goes back to Ohm's law. All right, Ohm's law, again, is the key. I told you would see this over and over and over again. All right, so let's take a look at, is everyone okay if I erase this middle part? Okay. So we're going to talk about Ohm's law one more time. Actually, a couple more times. All right, so remember with Ohm's law, it's basically the same. The driving force equals flow times resistance. Okay? Driving force equals flow times resistance. Flow times resistance. Okay? Now think about garden hose physiology. We're talking about this again in terms of blood flow. So it's almost a little bit more intuitive. What is the driving force? What is the driving force? What would be the driving force for water within a garden hose? What pushes the water around? Pressure. It's going to be actually a pressure gradient, right? It's going to be a pressure difference. All right, so in this case, the pressure difference is mean arterial pressure. MAP means mean arterial pressure. Minus venous pressure. Venous pressure. That's the pressure difference. All right, that actually equals a flow. Now the flow pertaining to your heart is actually cardiac output. Cardiac output in mils of blood per unit of time, minute. It's a flow, a river of blood, basically. Sounds terrible. A flow, that's the flow. So before we get too far down the line, let me just give you some information about cardiac output. Cardiac output is actually equal to heart rate in beats per minute times stroke volume. Stroke volume in mils, I shouldn't put the S there, mils per beat. So the stroke volume is when your ventricles contract, it's the amount of blood that's propelled out of the heart. It's mils per beat. All right? So beats actually cancel out. That's actually a unit. And cardiac output is going to be in mils per minute. Heart rate is the number of beats per unit time. And stroke volume is the amount of blood that's propelled out of the heart with every beat. All right, so that's how you would calculate cardiac output. And then finally, resistance in the heart is total peripheral resistance. TPR. Total peripheral resistance. And you can think about it as, we've been talking about those smooth muscle cells. If you have a lot of vasoconstriction, your blood pressure is going to go up. It's going to be a higher resistance, right? That's going to be what your heart is actually having to work against, that total peripheral resistance, blood pressure. The more your vessels are contracted, the higher the resistance is going to be. It's basically the difficulty with which blood flows through vessels. All right, so this is Ohm's law again, right? This is how it pertains to the heart. All right, a couple of things that I do want you to know is, does everyone have this down? I'm going to erase it again. I just want to give you a few more details to help you like really put things together. And then I'm going to give you a little bit of time, actually, to do this or do the first part of it just to get going on it a little bit. All right, so there's a couple of things that I want to kind of help you out with. The first one is, how do you calculate mean arterial pressure? All right? We're going to go back to, let's go back to the first part of this equation again. All right? One thing that you need to know is venous pressure is usually 0. Okay? It's usually around 0, so we can actually take this out of the equation. That's why a lot of times you see it's actually missing from the equation. So how do we calculate mean arterial pressure? Mean arterial pressure is going to be the diastolic pressure. Now we know what that means. Diastolic pressure plus 1/3 of the systolic pressure minus the diastolic pressure. Mean arterial pressure has everything to do with that blood pressure cuff I was talking about. You have to know the diastolic and systolic pressures. It's going to be the diastolic pressure plus 1/3 systolic minus diastolic. All right, so let's put some numbers to that. We've already talked about, a lot of times an average reading, a typical reading is 120/80 millimeters of mercury. I'll let you calculate that. What would be the mean arterial pressure if this was your reading? Falling apart here. When you have the answer, just raise your hand. Yeah.
93?
That's right, 93. So in this case, mean arterial pressure is actually going to be 80 millimeters of mercury plus 1/3, 120 minus 80, and that's going to be about 93.3, correct? 93.3. All right, why do we use this 1/3 value? Why would we use 1/3 in the equation? Now, let me just give you a little more information. You use 1/3 when you're at rest, when you're just sitting on the couch. This bumps up to 1/2 if you're exercising. So why is it 1/3? Think about where this is relative to our numbers here, 120 over 80? Why is it 1/3? This is a tough one, actually. Yeah.
Is it to like weight the sum because you're not contracting as much as you are relaxing?
That's right. That's exactly right. So another way to say that, you've got the intuition, is the heart is in diastole longer than it's in systole. So the mean arterial pressure when the body is at rest is closer to diastole, is closer to the diastolic pressure than it is the systolic pressure. So again, the heart is in diastole longer than it is in systole when your body is at rest. When you start to exercise, you can see how the mean arterial pressure, if this was 1/2, would be right in between the two values, more like 100. Okay, so that's how to think about mean arterial pressure. All right, one more thing. Let's talk about cardiac output one more time. We learned that cardiac output is heart rate times stroke volume. I actually had a friend who was a grad student with me, and she works at Medtronic now. She swears she got the job at Medtronic because in her interview, they started talking about cardiac output, and she said she blurted out, "Cardiac output equals heart rate times stroke volume," and they were like, "Yes!" Okay, so anyway, I think that's a really funny story. All right, so what is cardiac output? Stroke volume times heart rate. Now, you don't really have heart rate on here. What do you have? You actually have an R to R interval. R to R interval. So what we're going to be learning, some people that have the lab already understand this, but some people that don't have a lab are not as familiar. So let me just kind of show you what this is. If you actually, we're going to be learning about the ECG, the electrocardiogram, on Tuesday when we get back. The ECG looks something like this. You'll have different heart waves, the P wave, QRS, and T wave. P wave, QRS, and T wave. Now, some students have already measured their own waves. We'll learn that the P wave is actually atrial depolarization. QRS is ventricular depolarization. And T wave is ventricular repolarization. Don't worry that you don't understand these heart waves yet. Again, we will go over that. I just want to make sure that you know how to do the calculation. If for some reason you're bored over spring break and you want to do your sheet, I know everybody wants to do that. Okay, so this is the R to R value. The electrical currents sweep through the ventricles first, and then the ventricle contracts. Beat. Electrical current sweeps through the ventricles first, and then it contracts. So this technically, the R to R value, is 1 heartbeat, the interval, right? The interval. And the interval in this case is 800 milliseconds. Or I'm going to write this another way. 800 milliseconds is 0.8 seconds, right? You with me? 0.8 seconds. So 1 beat is going to be heart rate is equal to beats per minute, 1 beat every 0.8 seconds, right? 1 beat every 0.8 seconds. Does that make sense to everyone? So, to get heart rate, I would just say 60 seconds in 1 minute. What's the heart rate? now. What's the heart rate? 75 beats per minute. So, are we kind of putting everything together now? Here's what will happen on an exam. All right? You'll have to calculate mean arterial pressure. How will you do that? You'll calculate it because I will give you the systolic and diastolic pressures, and I'll tell you, is the person at rest or are they exercising? Okay? You'll have to calculate cardiac output. Okay? Cardiac output is heart rate. I'm going to give you the R to R value. And stroke volume is the one piece that I missed. Stroke volume is actually equal to the end diastolic volume. This is the amount of blood that's in the ventricle when it's completely relaxed. This will be the greatest amount. This will be the most blood. Minus the end systolic volume. This will be the amount of blood that's left in the heart when the ventricles are completely contracted. So I'll have you calculate cardiac output, heart rate times stroke volume, and then from there I may say, from what you know with your calculation about mean arterial pressure and your calculation of cardiac output, what is total peripheral resistance? So in that case, you would just divide the mean arterial pressure by cardiac output, and that will give you total peripheral resistance. Okay? And that's the extent. You can pretty much do all the questions on the sheet now. Pretty much every single question until you get to 7 and 8, basically. The back, there are some questions on the back. Ejection fraction is basically the stroke volume over the end diastolic volume. It's how much blood is actually propelled out with systole. Usually ejection fraction is about 55%. So it's going to be a percentage. Yeah. Yeah, so this is ejection fraction. Ejection fraction. It's basically the amount of blood that's forced out of the heart over the total amount of blood that enters in at the end of diastole when the heart is completely relaxed. It's the maximum amount of blood that's forced into the ventricle before it contracts. So if your heart, if you're in the clinics and you see, oh, ejection fraction is only like 20%, 15%, then you know that patient, whether it's a cat or a dog or a human, is probably in like heart failure, right? Because it's not giving that, real contraction, that contractility that it usually would. All right, so we do have about 15 minutes. Do you want to take, a good 5 minutes, look over the questions, ask me questions if there's something that you're still unsure about? And I just want to, maybe give you a chance to kind of look to see what these equations are all about. And then we'll start with the intro to cardiovascular, and I'll leave it with that. So take a look at the questions. Let me know if there's something that you really aren't understanding still. We'll try to clear it up. And again, don't worry if it doesn't gel right away. The more practice you get, the easier these get. Just like any of these problem sets, just takes a little bit of practice. Oh yeah, and don't worry about C lecture 9, slide 28. I thought I took that out, but it reappeared. When it comes to Ohm's laws, I do want you to memorize that. It's good. I'll tell you that too. It's good walking around knowledge. And that was done kind of. Yeah. Okay, let me just address the class. Okay, I know this seems very daunting at first, but Lean, can I actually borrow that really quick? Maybe I'll put it up on the document camera. She's done a very good job of adding to the equations for exam 2. Okay, so what you'll notice is in black at the very top are the equations that you're going to be given when this comes up. Sorry, there it is. So the equations are the mean arterial pressure is going to be given. You'll learn about Quasi-Hughes's law and net filtration pressure a little bit later. But you'll notice that cardiac output equals heart rate times stroke volume. Ohm's law is not on there. I do want you to memorize that because it is good walking around knowledge. I'm just going to say that. You should know Ohm's law. You should know how to calculate cardiac output, okay? And stroke volume, basically, and ejection fraction. Those are things that you should probably know. And I know that, so Lean did a good job of like filling in a lot of those blanks, but that is something I do want you to know for the exam. That won't be given to you. Okay, so take a look at that equation sheet again, figure out what's missing, and then make sure that you know those equations. Again, Ohm's law is mean arterial pressure equals cardiac output times total peripheral resistance. And to calculate cardiac output, that's heart rate times stroke volume. All right. So let's continue on. I know I gave you about 5 more minutes, but I just wanted to give you kind of a head start on what this calculation sheet is all about. This won't be due until the Thursday after you get back. So again, if you are going to some wonderful location and you have no books, you can pretty much catch up when you get back. Okay, so this will be due Thursday after we get back. I believe that's the 19th of March. All right, so just filling in some of the blanks. Let's go back to, for those that are home, we're going to go back to our sheet here. There we go. I'll put the screen back down. Again, this is our Ohm's law chart that's actually in your syllabus. What we're talking about now is Ohm's law in hydraulic terms. Flow is going to be mils per minute, some kind of unit of volume per minute. The driving force is the pressure change, the difference, the pressure difference, the gradient. And resistance factors are viscosity, vessel radius, and vessel length. Okay? All right, so let's talk about circulatory systems in general. We have about 10 more minutes. Circulatory systems move fluids by increasing the pressure of the fluid in one part of the body. The fluid flows through the body down its pressure gradient, always from high to low. It's the same with air as well. Air is always going to flow from high pressure to low pressure. And there are three main components, okay? The main components are you actually have to have a pump. The heart is our pump. Or propulsive structures. So sometimes there's auxiliary hearts in some, like insects. Or you have pulsing blood vessels due to smooth muscle cells, okay? So any kind of pump or propulsive system is required. A system of tubes is also required. We'll talk in just a minute about open and closed circulatory systems. And then finally, fluid. Fluid that circulates through the system is also important. Do you need hemoglobin? Some animals have hemocyanin. So hemoglobin isn't one of these critical factors. That is very important, obviously, for mammals. But other animals actually have a different type of molecule that carries oxygen, hemocyanin. So the three main components, a pump, a system of tubes, and fluid. What are some types of pumps? Basically, our heart, chambered hearts, are our pumps, our major pumps. We have atria that are reservoirs and the ventricles that are the actual pumps that propel the blood. Maybe you knew this, maybe you don't, but our skeletal muscles also act as pumps. So our skeletal muscle, think about your quads. When they contract, they actually help to propel blood from veins back to the heart. It helps with venous return. So one tip for you, if you're at graduation this year, don't lock your legs, right? You could, right? Bend your knees a little bit, contract those quads. It helps with venous return. Otherwise, you might not get enough blood to your brain and you may faint. It's called syncope, okay? So your skeletal muscle actually helps to move your blood through your circulatory system. Some animals have those pulsating blood vessels called peristalsis. Those are due to smooth muscle cells that surround those vessels. And it's also interesting that veins have one-way valves to ensure that the blood keeps moving in a one-way direction. If you're something wrong with your valves, especially in your legs, has anybody ever seen like varicose veins, right? Where those are problems with these one-way valves. Okay. All right. So there's different types of animals. This is a phylogenetic tree. A lot of you may have been taking or have taken zoology, and you probably learned all of the different kingdoms, right? There are animals that have an open circulatory system. We as mammals actually have a closed circulatory system. So you can also see that mollusks like snails, right, clams, these mollusks actually have an open circulatory system except for cephalopods like squid and octopi. Also, arthropods have an open circulatory system. So let's focus on the arthropods for a second, just so that you get some terms down. An open circulatory system has a large cavity where fluid actually can accumulate, okay? So these open cavities are, I think you can see this in pink here. It's just one large cavity. Fluid is drawn into these openings called ostia. And then you have these auxiliary hearts and hearts along the vessels that keep propelling the fluid up into the wings and then back into the cavity. Okay, so the cavity itself in an open circulatory system is called a hemocoel. Right? That's referring to the cavity, open circulatory system. the fluid that's circulating around is called hemolymph. Hemolymph. All right, and then again, you have your pumping structure. You have your set of tubes, even though it goes into an open cavity, and you have fluid. All right, so let's take a look at this. We only have a few more minutes. Basically, this is a comparative study looking at fish, amphibians, and the mammalian heart. So fish only have 2 chambers. Don't know if you knew that. Amphibians have 3 chambers. And mammals actually have 4 chambers to their heart. We actually have 2 atria and 2 ventricles. All right, so the fish heart, again, only has 2 chambers. The pacemaker cells are actually in the sinus venosus. Amphibians have 3 chambers. A lot of reptiles only have 3 chambers too. So take a look at this. Basically, they have 2 atria and 1 big ventricle. And so there's a lot of mixing of oxygenated blood and unoxygenated blood. Just really quickly, basically crocodiles and alligators are a lot more different than people once thought. Their morphology looks a lot of the same, but crocodiles now are thought to be more closely related to birds than alligators. So we'll talk more about this later, but crocodiles actually have 4-chambered hearts, just like birds and mammals do, okay? Whereas alligators only have the 3 chambers in their hearts. So you can see already that when you really dive into their morphology, crocodiles really are a lot closer to birds than they are alligators. Crocodiles also have a respiratory system where they are constantly getting fresh air. They have these posterior and anterior air sacs, and very much more like birds than they are alligators, which is interesting. All right, I think that I am running out of time. So I'm going to stop it there, but if you want to get ahead, like I know you all do, I would take a look at the anatomy of the mammalian heart. All right, so here's where we left off. Take a look at this, and I will have a game on Tuesday when you get back. Yay. Okay, I'll bring prizes. So have a wonderful spring break, everyone, and definitely be safe. All right, and I will see you when you get back Tuesday, March 17th. As you're leaving, also, don't forget to pick up your homework assignment as you're leaving.
So muddy and puddly, and we were jumping around.